Hello everyone. Today we are going to discuss chemistry of carbohydrates. So what is the definition of carbohydrates? Okay. The definition of carbohydrates will be carbohydrates are polyhydroxyaldehyde or polyhydroxyketone, or they are condensed products or their derivatives. So basically the carbohydrates are compounds where obviously there will be many carbons. The functional group will be aldehyde or ketone, and then there will be multiple hydroxyl or OH groups, as we will see later in their structures. So they are polyhydroxyaldehyde or polyhydroxyketone, and or it can also be something that is a product of such polyhydroxyaldehyde or ketone when many of them are condensed together. So this is the definition. The empirical formula, the general formula of the carbohydrate is like CnH2ON. The N is the number of carbon atoms. So it can be C6H12O6, that is C6H2O whole 6, which is the formula for glucose, as you all know. Okay. So next let us go to the classification of carbohydrates. Okay. Now carbohydrates can be classified depending on different factors. The main factor, the main kind of classification is depending on the number of saccharide or the sugar units. That is the definition that we said, that it is the polyhydroxyaldehyde or ketone. So the number of such polyhydroxyaldehyde ketone molecules depending on that. So how can it be classified? It can be monosaccharide when only one such unit is there, disaccharide when two such units are condensed, oligosaccharide, oligo means some, so when 3 to 10, up to 10 we will call it oligosaccharide, and then if it is more than 10 then we will call it polysaccharide. Poly means many. Okay. So the thing is, if you get a monosaccharide, then there is only one polyhydroxyaldehyde ketone or saccharide or carbohydrate unit. So if you hydrolyze it or break it down with water, there is no further carbohydrate you can get. So that is only single unit. So similarly, disaccharide means if you break the bond, you can break it to two monosaccharide units. And in case of polysaccharides, if you can break all the bonds that has joined or condensed them, then you will get multiple such monosaccharide units. Okay. Now monosaccharides, now first we will discuss the monosaccharide, the basic unit of carbohydrate, that is the one carbohydrate unit. So how can we classify monosaccharides? The monosaccharides can be classified depending on mainly two characters. Number one, what is the functional group? We have seen it can either be aldehyde or ketone. So depending on that, or depending on what is the number of carbons in each of the monosaccharide unit. Okay. So here we can see that if the functional group is aldehyde, that is CHO, you must have learnt about it in chemistry. So then those monosaccharides will be known as aldose. And if the functional group is ketone, that is C double bond O, then those monosaccharides will be known as ketose. And then depending on the number of carbon in that particular monosaccharide, it can be, if it is 3, then triose, 4 carbon, tetrose, 5 carbon, pentose, 6 carbon, hexose, and 7 carbon, heptose, like this. And this can be continued, but most of the naturally occurring important carbohydrates are within this range, 3 to 7 carbon. So if an aldose has 3 carbons, then it is aldotriose. So it is aldose as well as triose. Similarly, 3 carbon ketose will be ketotriose, and similarly there are names. So 6 carbon aldose will be aldohexose, and 6 carbon ketose will be ketohexose. The 6 carbon hexoses are most common of all the carbohydrates. So in red color you can also see the examples of each group. So for example, aldohexose, 6 carbon aldose sugars are glucose, galactose, mannose, whereas ketohexose or 6 carbon keto sugar example is fructose. Aldopentose example is ribose, xylose, arabinose. And ketopentose is example is ribulose, xybulose. Okay. So let's see two examples. One aldo sugar, that is glucose. You can see 6 carbon and CHO at one end is the functional group. And ketohexose, the typical example, the most common example we give is fructose. You can see again 6 carbon, but the functional group is at carbon 2, that is CO, C double bond O. Okay. So before going into more detail of their structure, let us now discuss another important thing, that is stereoisomerism. The carbohydrates, they show a very important isomerism property that is known as stereoisomerism. So what is stereoisomerism? Stereoisomerism is a character or isomerism that is due to the presence of asymmetric carbon or the chiral carbon. Asymmetric carbon means a carbon where you know that carbon atom has 4 covalent bonds. It can form 4 covalent bonds. So if a carbon with its all 4 covalent bonds forms with 4 different groups, then that carbon is known as an asymmetric or chiral carbon. Okay. So for example, in this glyceraldehyde, we can see the second carbon, the carbon at the center. This carbon has 4 different groups attached to it on the 4 covalent bonds. The other 2 carbons, the first carbon you can see C double bond O. So 2 covalent bonds together with O. So it is not asymmetric. The third carbon you can see CH2OH. So 2 hydrogen are attached. So again, this is not asymmetric carbon. But the second carbon, the carbon at the middle, it has 4 asymmetric carbons. The 4 covalent bonds are bound to 4 different groups. So around this asymmetric group, if the orientation of the groups are different, then that kind of isomers are known as stereoisomers. So here, as you can see, the glyceraldehyde, which is the smallest monosaccharide, is taken as the standard. So if the OH group is on the right side, then it is known as D-glyceraldehyde, D isomer. And if the OH is on the left side, as has been pointed by the red arrow, then it is L isomer, L stereoisomer, then it is L-glyceraldehyde. So similar examples, similar things, D and L isomer will be discussed when we will go to the more big or larger carbohydrates like glucose, fructose, etc. Okay. So if we want to define it, that stereoisomers are type of isomers which have same chemical formula, same molecular formula. The molecular formula is same. The D and L glyceraldehyde, you can see the formula is same, but the structure differs in the spatial orientation of groups. That is orientation of groups around space, around the asymmetric carbon atom. So if there is any asymmetric carbon atom, and then if the groups around that asymmetric carbon atom are different in their orientation in space, then those isomers are known as stereoisomers. Okay. So without getting into too much detail of that, let us come to glucose again, which is the most common carbohydrate and which you need to know a little bit more about. So what are the different types of isomers you can get in glucose? So in glucose, the stereoisomer, different kind of, it can first be the functional group or structural isomer that we know. So the functional or structural isomer of glucose will be, if it is aldo sugar, then glucose, and if it is keto sugar, then it is fructose. We all know. So these are functional and structural isomer. Okay. But next, what the topic that we are trying to focus upon is the stereoisomers of glucose. Okay, so let's again come back to stereoisomers. So stereoisomers, we have said, are having same molecular formula but different in the orientation of groups in space. Okay, so in case of glucose, we will see there are three types of stereoisomers we can find. First, the D and L glucose, D glucose, L glucose, which is the in comparison with the D-glyceraldehyde and L-glyceraldehyde, as we have seen. Then there are some other types of stereoisomers known as anomers, apimers, etc. So let's just discuss them little bit. Okay. So here we see the D-glucose and L-glucose. So here there are, in glucose, there are 4 different asymmetric carbons. The CHO is not asymmetric on one end and CH2OH is not asymmetric on other end. But the 4 carbons at the middle each have HOH on 2 sides and other groups on other 2 sides. So they all 4 are asymmetric. So the last one of them, which is marked by yellow, that will be taken for D and L isomer. So if the OH is on right side, it is D glucose. If OH is on left side, it is L glucose. Okay. In these two, the D and L isomer, they are also known as enantiomer. Now in these enantiomers, since they are actually same molecule, so their physical and chemical properties are same. But only one property differs, and that is optical rotation. We will come to it little bit later, but optical rotation is if there are some molecules which have such enantiomers or stereoisomers, then if you pass certain kind of light which is known as plane polarized light, then that polarized light rotates towards right or left. That characteristic will differ between the stereoisomers. In human body, in nature, the usual form of sugars are in the D form. So glucose in our body, usually we find it in the D glucose form. That is the naturally occurring form. Okay. Between these D and L isomeric forms, they can be interconverted by some enzymes. The D isomer and L isomers can be interconverted, and that enzyme is known as racemase. So racemase can interconvert from D to L or L to D. Okay. So the next kind of isomer is known as anomer. In glucose, we will, or in any sugar, we will find 2 different types of anomers, that is alpha and beta glucose. The alpha and beta, these 2 forms, they are due to spatial orientation difference at the carbon number 1 or the functional carbon. Now you may say that the functional carbon is not asymmetric carbon. So how can there be, as for our preliminary definition, there cannot be stereoisomer. It has to be an asymmetric carbon, but first carbon was not asymmetric. But yes, it can be asymmetric. How? Okay. So next, here we can see that in solution, the first and fifth carbon, as you can see, can join together. And when they join together, you can see they form a ring-like structure. And then the previous carbon number 1 enters into the ring, and that's why it becomes an asymmetric carbon. As you can see here, in the last 2 structures, the HOH is attached to the carbon number 1, and if the OH is above the plane, as marked by red, you can see, then it is known as beta isomer, and if it is below the plane, then it is alpha isomer. So the D glucose in solution can form a ring-like structure and then form 2 other isomers, that is beta D or alpha D glucose. Similarly, L glucose can also form alpha L and beta L glucose, but since naturally occurring glucose or sugars are mostly D form, so we will focus on D form. Okay. Next, in this structure, again, this is the ring structure. The carbon number 1, we can see, now has entered into the ring and become an asymmetric carbon. Now if OH is below the plane, then it is alpha glucose, and if the OH is above the plane, then it is known as beta glucose. Okay. So what is anomerism? So this kind of isomerism is known as anomerism. The anomers of glucose are not stable by in that form exactly in alpha and beta, and they often interconvert from alpha to beta and beta to alpha. The ring opens and again form, opens and again form, and alpha becomes beta, beta becomes alpha. So this anomerism, this property is interconversion, and this interconversion property is actually known as mutarotation. So mutarotation term is used for this interconversion between alpha and beta isomeric forms, mutarotation. And this kind of isomers are known as anomers. Okay. So the next type of isomer that we discuss is epimer. So what is epimer? So in the first structure, we have seen when we discussed the D and L isomer of glucose, we have seen that there were 4 asymmetric carbons, but only the last one we have considered for D and L. So the position can also differ in the other 3 asymmetric carbons also, that is C2, 3 and 4. So if 2 isomers are different in spatial orientation around those other asymmetric carbons, then they are known as epimers. So there are 2 common or important types of epimers of glucose. One is galactose, another is mannose. So galactose is called C4 epimer because the C4 is different, and mannose is known as C2 epimer. Okay. So in this structure here we can see that you can see focus on carbon number 4 between glucose and galactose, and you see the orientation is different at carbon number 4. And similarly in mannose, you can see the orientation of groups are different in carbon number 2. Okay. So these are glucose and mannose are anomer, glucose and galactose are anomers of each other. Okay. So next we come to optical activity. What is optical activity? I have already mentioned that when there is any isomers, stereoisomers, and if there is asymmetric carbon, if you pass a plain polarized light through it, then the light will rotate, and that property is known as optical activity. And those solutions are known as optically active solutions. So glucose, obviously it has DL isomers, so it is optically active. So if you pass plain polarized light through it, then the light can either turn towards left or towards right. If it turns toward right, then that isomer, that optical activity is known as dextrorotatory or small d. The isomers were capital D, capital L. And if the light rotates towards left side, then that property is known as levorotatory optical activity or small l. So D and L, dextro and levo rotation, also is designated with plus and minus sign respectively. So dextro rotation, you give sign plus x degree, or if it is levo rotation, then minus x or y degree. Okay. Now let us come to some of the chemical properties of monosaccharides. One of the very important property of monosaccharides is its reducing property. So all these monosaccharides, glucose, mannose, galactose, fructose, in the alkaline medium, they convert into a form known as NADol. And this NADol is a very, very strong reducing agent. And we can test this by reacting the NADol with the cupric ion, which will then be reduced to cuprous ion. Okay. And this property can be tested by 2 very common tests known as Benedict test and Fehling test. Okay. So next, what is the most important function of glucose in our body? Glucose is a primary source of energy to all the body cells. So all the body cells that continuously need much of energy, the fuel that it uses is glucose. The fetus during the intrauterine life also, the glucose is the main fuel. And some of the tissues like brain, RBC, lens, spinal cord, peripheral nerves, they are almost completely dependent on glucose. So they can only use glucose as fuel and nothing else. Some other cells, they can although use some other fuels like fatty acid, etc. But these cells like brain, erythrocyte, lens, etc., they almost completely depend on the glucose for their energy. Okay. When we are in the well-fed condition, when we have eaten. So then we have excess amount of glucose in our body. So that excess glucose we store in our body for the time when we are not eating, and we can then supply from that store. So how do we store it? In plants, the excess glucose is stored as starch, and in animals, it is stored as glycogen. And when the glucose is even much more, then you can store as glycogen. It can then be converted to fat and then triacylglycerol, and then it is stored like that. Okay. Glucose can also be used for the synthesis of a lot of many important substances like glucuronic acid, glucosamine, N-acetylglucosamine, galactose, some amino acids, some glycosides, etc. We'll not go into much detail of them, but there are many important molecules that can be synthesized from glucose. Okay. So with this discussion, we finish the discussion of monosaccharide. Now we can move to disaccharide. So what are the disaccharides? They are the carbohydrates where there are two monosaccharides are joined. And how they are joined? They are joined by a particular type of bond known as glycosidic bond. Okay. So what is glycosidic bond? It is a bond when two molecules are joined through two hydroxyl groups. But one of the hydroxyl group has to be a monosaccharide anomeric carbon OH. Okay. So two OH groups of two monosaccharides joined together. So two monosaccharides joined together by removal of one water through interaction of two hydroxyl or OH groups. But one of the OH must be from the anomeric carbon. The anomeric carbon we have seen, that is the carbon that forms after the ring formation. Okay. So as the anomeric or functional carbon participate in the glycosidic bond formation, so that will no longer be free, and the reducing property was due to that group, so it will lose its reducing property. So the monosaccharide OH group that involves in the glycosidic bond formation is no longer available for the reducing property. Okay. So accordingly, disaccharides can be reducing or non-reducing disaccharides. So reducing disaccharide means that among the two monosaccharides, one has given the anomeric carbon, which it must. The other one's anomeric carbon is still free among the two monosaccharides. So the examples are lactose, maltose, isomaltose, lactulose, cellulose, etc. You know lactose is found from milk, and maltose is malt sugar, etc., etc. These things you know. Okay. So similarly, there are some non-reducing disaccharides where both the anomeric groups of both the monosaccharides are involved in the bond formation. So its most important example is sucrose, which is the cane sugar, which is the common sugar that we use. Okay. So among the disaccharides, if we see one by one, some of the important ones, lactose is the reducing disaccharide where one galactose and one glucose are joined together. Lactose is formed by one galactose and one glucose. Beta D galactose and beta D glucose together will form the lactose, and the bond is beta 1,4 glycosidic bond. So carbon 1 of galactose, that is the anomeric carbon, is involved in the bond, but in case of glucose it is the carbon 4. So the carbon 1 or anomeric carbon of glucose is still free, so it can still show reducing property. Then there is maltose. It is also a reducing disaccharide. It is joined by two glucose. Lactose was galactose plus glucose, but maltose is glucose plus glucose joined through alpha 1,4 glycosidic bond. Okay. So this is the structure how they join, but you may skip this. Okay. So where we find maltose, it is found in malt grain, germinating seeds, etc. And in our body, usually we get maltose from digestion of starch and glycogen, that is through our food source, the plant source usually starch, animal source glycogen, and then it is digested by alpha amylase enzyme, and then it produces maltose. Okay. The next one is sucrose. We have discussed it is a non-reducing disaccharide. It is called the cane sugar, the common sugar we use. Okay, so it is formed by one glucose and one fructose. The components are alpha D glucose and beta D fructose, and the bond is, this is very interesting. See this, that it is alpha 1 beta 2 glycosidic bond. So the carbon 1 of glucose, which is the functional carbon, and the carbon 2 of fructose, which is again the functional carbon, both are involved in the glycosidic bond. So none of them are available, so it is non-reducing. Okay, so this is the thing, that glucose and fructose, both the anomeric carbons are involved, so it is non-reducing. Okay, so the three most important disaccharides that we discussed are lactose, maltose, and sucrose. Lactose is galactose plus glucose, maltose is glucose plus glucose, and sucrose is glucose plus fructose. Okay, so the next, from the monosaccharides and disaccharides, we can now go to oligosaccharides, which are formed by joining of 3 to 10 monosaccharide units. More than 10 we will call poly. Okay, so the next most important thing is polysaccharides. Polysaccharides means more than 10. They are also called glycans. Polysaccharides are also called glycans. Okay, so what is polysaccharides? Polysaccharides are complex class of carbohydrates where there are more than 10 monosaccharides. The 10 monosaccharides can be same or can be different. So same type of monosaccharides or different types of monosaccharides. And they repeatedly, one after the other are joined through successive glycosidic bonds. Okay, so monosaccharides can be, number one, homopolysaccharides. Homo means same. Homopolysaccharides. So if all the more than 10, it can be 100, can be 1000, can be 10,000. So all of them, if are composed of same type of monosaccharides repeatedly, then it is called homopolysaccharides or homoglycans. We have said that polysaccharide or glycan, homopolysaccharide or homoglycan. Okay, some of the common examples of homopolysaccharides are starch, glycogen, cellulose, and inulin. Okay, so starch is, we have said that there is the storage form of glucose in plant, and it is joined repeated unit of alpha D glucose. So alpha D glucose, alpha D glucose, alpha D glucose, like that. Okay, so starch can be of two types. The first type of starch is amylose. Here it is joined through successive glucose in a straight chain, one after the other with successive alpha 1,4 glycosidic bonds. About 20% of all starch is in this form. Okay, the next is amylopectin. The another form is amylopectin. This is more 80 to 85% in this form. Here what happens, there are some branch points. So the straight chains are same as amylose, alpha 1,4 bond, but there are some branch points with alpha 1,6 bonds. Okay, so in this diagram you can see that there are some straight chains and then there are some branch points, and you can see how they are mentioned, that all the straight chain bonds are alpha 1,4 bonds, whereas the alpha 1,6 bonds are at the branch point, and it is highly branched and very, very large and big molecule it can be. Okay, some of the common sources of starch are all the plant sources, as it has been from the different seeds and rice and wheat and potato and beetroot, etc., etc. all are sources of starch. Okay, so it obviously, as we have discussed, it stores energy in the plant, and in human it is the most important source of supply of carbohydrates. So all we get are carbohydrate mainly from rice and wheat and potato, etc. So like that. Okay, how do we digest starch in our body? The enzyme is amylase, as we have said previously. The amylase can be salivary amylase, and most importantly, the most of the starch is digested in the intestine by the pancreatic amylase. And they break all those alpha 1,4 bonds and produce maltose and isomaltose. Okay, and then maltose, isomaltose will be further degraded by maltase and isomaltase enzyme, and so finally starch is broken down into large number of glucose molecules, which we then absorb and our cells use. Okay. The next thing is glycogen. So glycogen is the same thing like starch. It is also a homopolysaccharide, also again made up of alpha D glucose joined by alpha 1,4 bonds, but it is the glucose source. This is the glucose storage form in animals. Okay. So the glycogen entirely is in the branched structure, which is similar to the amylopectin. No linear form like amylose, only branched like amylopectin, but it has more branches than starch amylopectin. Here the branches, every 8 to 10 glucose units you will find a branch with alpha 1,6 bond, whereas in starch you will find, in amylopectin you will find branch much less. Every 24 to 30 residue later you will find a branch, but here every 8 to 10 residue later you will find a branch. Okay, so this is the structure. You can see there is much more branch and more compact structure. Okay, so in animal body glycogen is found mainly from two sources, liver and skeletal muscle. These are these two areas where glycogen is formed and stored. Okay, so when we are in the well-fed condition, when we have taken a large carbohydrate meal, the excess glucose will be stored as glycogen, and that process will be known as glycogenesis. And then we need that glucose, most importantly during fasting, when we are not eating, say 4, 5, 6 hours has passed since we have eaten and now we are not eating. So now we need glucose from our storage. Then that same stored glycogen will be broken down, and that process will be known as glycogenolysis. Okay, so these processes we will discuss when we discuss carbohydrate metabolism. Okay, so the dietary glycogen again will be metabolized same way as the starch by the amylase enzyme, and then from maltose and isomaltose by maltase and isomaltase enzyme. Okay, the glycogen breakdown in liver, that is liver glycogenolysis, it is important for maintaining the blood glucose level during fasting. When we are not eating, our blood glucose level falls, and where from that glucose will come and maintain the glucose level? It is the glycogen that was stored in the liver after we have taken a meal. So that stored glycogen from liver comes and continuously maintains blood glucose level. Okay, but in contrast, the glycogen that was stored in muscle, it will not help to maintain blood glucose level, but it is utilized only for the muscle activity, that is the muscle contraction. So that is the difference. Liver glycogen maintains blood glucose for other tissues, and muscle glycogen, the glycogen that is stored in muscle is used only for its own use during the muscle contraction. Okay. Next type of homopolysaccharide is cellulose. The cellulose is a homopolysaccharide that, unlike starch and glycogen, we cannot digest. This kind of polysaccharide we cannot digest in our intestine. So it remains in the intestine and it forms a particular very important source of diet that is known as dietary fiber. Although it is not absorbed, still it is an important thing. Why? We will come to it later. Okay. So what is this cellulose? It is again glucose, but it is beta D glucose. Starch and glycogen was alpha D glucose, but it is made up of multiple beta D glucose, beta D glucose, beta D glucose, beta D glucose joined by beta 1,4 glycosidic bond. So starch and amylase, it was alpha D glucose joined by alpha 1,4 glycosidic bond, but here it is beta D glucose joined by beta 1,4 glycosidic bond. Okay. So where do we find cellulose? It is very abundant in the plant universe. It is large amount of all the fibrous things, they are all made up of cellulose. So plant cell wall, actually the cell wall of plant, they contain cellulose. So you can, in what kind of diet we can find? So in the outer covering of the grains, green leafy vegetables, cabbages, spinach, nuts, beans, all the vegetables, fruits, their peels, etc., you will find a large amount of cellulose. Okay. So as I've said that this cellulose in our diet is not digested, not absorbed in our intestine. The enzyme that digests it is called cellulase, like amylase that breaks the starch and glycogen. It is cellulase, but we do not have that enzyme. So we cannot break it. We do not get any calorie. So what is its purpose in our diet? It serves as dietary fiber. dietary fiber. So what is the role of dietary fiber in our diet? Okay. So since it remains in the intestine, it acts as roughage. It draws a lot of water, and therefore the overall volume and bulk of the stool is increased. So more fiber you take, the bulk of the stool will increase because it will take more water and the stool will become soft. So there will be much less constipation. The stool will pass much easily through the intestine. So the most important role of the dietary fiber is to form the volume and bulk and softness of the stool so that it can pass easily through the intestine and constipation does not occur. So if somebody takes a very low fiber diet, he or she will definitely suffer from constipation. Stool will become hard, the stool volume will become much less, and the stool will not pass easily. But apart from that, it also has some other very important roles. That it has been seen that it helps to prevent colon cancer and hemorrhoids, and it has some other very important roles also. Okay. For example, since the cellulose does not get digested and absorbed, it does not give any glucose or calorie to your body, but since you have eaten a lot of food, so your stomach seems full, so your satiety is achieved, so you think you have eaten enough, so overall the calorie intake becomes less and less, so the risk of obesity decreases. Okay. So after the homopolysaccharides, now let us move to the heteropolysaccharides. So obviously from definition we can understand in heteropolysaccharide there will be more than 10 monosaccharides, but they will be not only of one type but different types of monosaccharides. They does not need to be 10, 12, 13 types of monosaccharides, but even if it is just two different types of monosaccharides, then again we call them heteropolysaccharides. So more than 10 units, but more than one type, not just same type all of them, but they can be different types, maybe 2 types, 3 types, 4 types of different monosaccharides, and they are again linked by glycosidic bonds. Okay. So the first thing that we need to discuss among the heteropolysaccharide, most important, is glycosaminoglycans. So glycosaminoglycans are the first important heteropolysaccharides. Glycosaminoglycan or GAG. So how they are made up of? They are made up of two different types of things, uronic acid and amino sugar. Uronic acid and amino sugars are derivatives of monosaccharides, and there are repeating units of this. So uronic acid, amino sugar, uronic acid, amino sugar, uronic acid, amino sugar, maybe for 100 times. So it is a polysaccharide and heteropolysaccharide. Okay. So what are the uronic acids that can be found in GAG? Glucuronic acid or idiuronic acid. And what are the amino sugars? Glucosamine and galactosamine. Actually N-acetyl glucosamine and N-acetyl galactosamine. Okay. So what are the common properties of GAGs? The common properties of GAGs that the GAGs show, all of the GAGs, they show some common properties. That GAGs have some negative charge groups like carboxyl groups, and then sulfates and phosphates are attached. So they draw a lot of water. They can draw a lot of water. Okay. So because they have a lot of water and because they have negative charges which repulse each other, so they become a slippery or sticky kind of thing, and which feels like little bit like mucus. So they are also known as, the GAGs are also known as mucopolysaccharides because they get a feeling like mucus, a slippery sticky kind of feeling. So they are also called mucopolysaccharides or MPS. And because it can hold a lot of water, so it can take a large volume together. Okay. So the mucopolysaccharides or glycosaminoglycans, MPS or GAGs, the common physical property is high viscosity, high density, tartar or stickiness, etc. These are the common features. Okay. So what are the common examples? What are the examples? There are 7 different types of GAG. Only 7 different types of GAG molecules. So what are their names? Hyaluronic acid, heparin, heparan sulfate, chondroitin sulfate, darmatan sulfate, and keratan sulfate 1 and keratan sulfate 2. So if you include keratan sulfate 1 and 2 separately, there are 7 different types of GAGs. Okay. So from GAGs, next we go to the next kind of molecule, which is known as proteoglycans. So what is proteoglycan? In proteoglycan, there will be a core protein which will then be attached with GAGs. So GAG attached with a protein forms a structure known as proteoglycan. Proteo means protein and glycan means the polysaccharide. So here it is what kind of polysaccharide? Heteropolysaccharide, GAG. And core protein and GAG will be joined through again glycosidic bond, which can be O or N glycosidic bond, but that structural detail you don't need to know. Okay. So in the proteoglycan, in this structure, proteoglycan, which is also called mucoproteins, here the carbohydrate part will be more than 10%. So carbohydrate is proportionately high. Protein is proportionately less and carbohydrate is more than 10%. Usually it can be more than 50, 60, 70%. Okay. So in this structure, you can see the blue thing is the protein. So it is the core protein. And then the red things, they are the GAG chains, like heparan sulfate, like it can be chondroitin sulfate, it can be keratan sulfate, etc. So the red chains of the GAG, so the chains of GAG are attached to one central core protein. So there will be a core protein at center and then many GAG chains that are attached to it. This is the structure of proteoglycan. Okay. So in this structure also you can see that there is a central core protein which is drawn with red, and then there are yellow and green GAG chains that are attached to it. And mostly these kinds of protein are found in the cell membrane. As you can see, the red protein goes through and through the cell membrane. Usually in the membrane protein, these kinds of things are found. Okay. So some of the examples of proteoglycans are given here, like agrican, biglycan, beta-glycan, decorin, sarglycan, syndecan, etc., etc. These are some of the names. You can remember few of them. Okay. So these proteoglycans and mucoproteins, they have more or less similar properties to the GAGs or mucopolysaccharides. So they are usually found in the cell membrane, in the extracellular matrix of the connective tissue like bone and cartilage. And they usually, their function is to provide a structural and mechanical support to that tissue. The proteoglycans, this is their common function. Okay. So next we go to the next type of molecule, that is glycoprotein. Glycoprotein. Okay. So glycoproteins again are proteins that are conjugated with carbohydrate. So the proteoglycan was protein plus carbohydrate. Here also it is protein plus carbohydrate. So what is the difference? Here the carbohydrate is not as large molecules as GAG. In proteoglycan it was large GAG molecules, GAG chains were attached, but here small oligosaccharide chains usually are attached. That means less than 10 carbon. Such oligosaccharide chains are attached, and they can be branched, they can be unbranched. Again, they are joined by glycosidic bonds. Okay. So here again the structure is, there will be a protein and then oligosaccharide chain will be attached. In proteoglycan we have seen the protein attached with GAG, here the protein attached with oligosaccharide chain. But here the difference is the carbohydrate content is much less. It is less than 10%. More than 90% if you take the molecular weight of the total thing, more than 90% is protein, less than 10% is carbohydrate. But in proteoglycan, protein is comparatively less and carbohydrate is much more. At least more than 10%, but usually it can go up to 50, 60, 70%. Okay. The glycoproteins can be divided according to the glycosidic bond that is attaching the protein and carbohydrate into N-linked glycoside and O-linked glycoside. In case of N-linked glycoside, the glycosidic bond is with the asparagine amino acid of the protein, and with O-linked it is the OH group of serine or threonine. But this part again you can remember. If you don't remember, then again it's okay. Okay. So what are the important glycoproteins? A very large amount of proteins in our body are actually glycoproteins. In fact, all the proteins that we find in plasma, most of the proteins, all those plasma proteins are glycoproteins in nature. Some of the other important proteins that can act as glycoprotein as their structure are collagen, which is a very, very important protein, very common protein, which is found in most of the tissues, in all the connective tissue, in bone, in muscle, etc. That collagen is also a glycoprotein. There are many, many important enzymes like alkaline phosphatase, ribonuclease B, and there are many other enzymes. They are also glycoprotein in nature. Okay. Then there are many other types of proteins. They are also glycoproteins. Some hormones are glycoproteins. Some hormone receptors are glycoprotein. Another very important membrane protein known as glycophorin is also glycoprotein. The mucin, that is the main thing in mucus, is also, that acts as lubricant, is also glycoprotein. So see, the mucin, that is the main thing in mucus, that is actually a glycoprotein. But the character is so similar with the proteoglycans that we were discussing and glycosaminoglycans. That's why glycosaminoglycans are also called mucopolysaccharides, but that is different. Mucin itself is a glycoprotein. Then there are many other transfer proteins, transport proteins, like transferrin for iron, ceruloplasmin for copper. They are also glycoproteins. So a large variety of proteins in our body with large variety of functions, all of them are glycoproteins. Okay. And then there are other things, glycoproteins, along with some other molecules known as glycolipids, which are lipid plus carbohydrate, that we will discuss when we discuss the chemistry of lipids. They all form a very important part of the cell membrane, which is known as glycocalyx. Then there are some clotting factors, there are some hormones, then immunoglobulins. So many different types of proteins in our body, all are glycoproteins. A large proportion of all the proteins in our body actually are glycoproteins. Okay. So thank you very much for your patient hearing. Thank you.